Welcome to the Bodhi Academy. My name is Amisha Praso, and in today's video we will continue with our chapter The Fundamental Units of Life, class 9, chapter 5. So previously in this video we started talking about the different examples of cell organelles. So we started by endoplasmic reticulum, Golgi apparatus and lysosomes. So talking about endoplasmic reticulum, we said that it is a large network of membrane bound tubes and sheets. So further we said that this endoplasmic reticulum which is there, it is further divided into RER and SER, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. In rough endoplasmic reticulum, we said that it appears rough due to the particles which are attached to the surface of the endoplasmic reticulum. Here this endoplasmic reticulum which is there, it has these ribosomes which are present in all the active cells and they help in the manufacturing of proteins. Once these proteins are manufactured, they are sent to various places as per the need with the use of the endoplasmic reticulum. The smooth endoplasmic reticulum, it helps in the manufacture of fats, molecules or lipids which are important for cell function. Some of these proteins and lipids, they help in building the cell, and this process is known as the membrane biogenesis. Some other proteins and lipids function as enzymes and hormone. Although the ER, it varies greatly in appearance in different cells, it always forms a network system. Also, we spoke about some of the functions of ER. First is to serve the channels of transport for materials between various regions of cytoplasm or cytoplasm and the nucleus. The ER, it also functions as a cytoplasmic framework, providing a surface for some of the biochemical activities that take place in the cell. In the liver cells of the group of animals called as vertebrates, it plays an important role in detoxifying the poisons and the drugs that are present. Next, we spoke about the Golgi apparatus. In that, we first said that it was described by Camillo Golgi, and it consists of membrane bound vesicles which are arranged approximately parallel to each other in stacks known as cisterns. These membranes, they often have connections with the membranes of the endoplasmic reticulum and therefore constitute another portion of complex cellular membrane system. The material that is synthesized near the ER, it is then packed and dispatched to various targets inside and outside the cell through this Golgi apparatus. Its main function is modification, packaging and storage of products in the vesicles. In some cases, the complex sugars which are there, they are formed from the simple sugars in the Golgi apparatus. The Golgi apparatus is also involved in the formation of lysosomes. Now, next we spoke about the lysosomes. So, lysosomes, we said, are membrane bound sacs filled with digestive enzymes. These are enzymes, they are made by rough endoplasmic reticulum. So these lysosomes are kind of a waste disposal system of the cell. So they help in cleaning as well, by digesting the foreign material as well as the worn out cell organelles. The foreign materials which enter the organelles, say maybe it can be food or bacteria, they are broken down from complex systems into simpler substances. The lysosomes, they are able to do this because they contain these powerful digestive enzymes which are capable of breaking down all organic matter. During any kind of disturbance in the cellular metabolism, for example, say that the cell gets destroyed. So in that case, these lysosomes, they burst open and the enzymes, what they do is that they digest their own cell. This is one of the reasons why lysosomes are known as the suicide bags. Now, in today's video, we will speak about mitochondria, plastids, vacuoles and cell division. So, mitochondria, it is known as the powerhouse or the energy provider. Now, let's see why it is called so. So, mitochondria are known as the powerhouses of the cell. The energy required for various chemical activities needed for life is released by this mitochondria. So any kind of chemical activity that is happening, it requires some kind of energy, and this energy is released by the mitochondria in the form of ATP. ATP is adenosine triphosphate molecules. So any energy that is required for any kind of chemical activities is released by the mitochondria in the form of ATP molecules. Therefore, ATP is known as the energy currency of the cell because it is providing energy for each and every chemical activity that is taking place inside our body. Mitochondria have two membrane coverings instead of just one. So till now all the cells that we have discussed, we said that they have one single membrane, but mitochondria has this two membranes. So the outer membrane which is there, it is very porous, while the inner membrane is deeply folded. Now these folds, what they do, they increase the surface area for this ATP generating chemical reaction. So the inner membrane, what we are saying, it is highly porous, and the outer one is having these deep folds, and these deep folds, they increase the surface area for ATP generating chemical reactions. Mitochondria are strange organelles in the sense that they have their own DNA and ribosomes. Therefore, they are able to make some of their own proteins. So that was about mitochondria. Next, let's see about plastids. So plastids, they are present only in the plant cells. Now there are two types of plastids: chromoplasts, that are the colored plastids, and the leucoplasts, that are either white or colorless plastids. So the chromoplasts are the colored plastids that are present in leaves, flowers and fruits. So the chromoplasts are the reason why these leaves, fruits and plants, they have color. Then plastids containing the pigmented chlorophyll are known as the chloroplast. They are important for photosynthesis in plants. So we know during photosynthesis chlorophyll is very important, and this chlorophyll pigment in plants is due to this plastid chloroplast. So chloroplast also contain various yellow or orange pigments in addition to chlorophyll. Leucoplasts are found primarily in organelles in which materials such as starch, oils and protein granules are stored. So leucoplasts, they are found in primary organelles in which materials such as starch, oils and protein granules are stored. The internal organization of plastids, it consists of numerous membrane layers embedded in a material called a stroma. Plastids are similar to mitochondria in external structure because even they have their own DNA and the ribosomes. Next we have the vacuoles, that is the storage. So vacuoles are the storage sacs for solid or liquid contents. So vacuoles are small sized in animal cells, while in plant cells they are very large vacuoles. So that is up to 50% to 90% cell volume. So in plant cell it is around 50 to 90, and in animal cell it is below this 50% of cell volume. In plant cells, vacuoles are full of cell sap and provide turgidity and rigidity to the cell. In amoeba, the food vacuole contains the food items that is consumed, and it contractile vacuoles expels excess water and some waste from the cell. So what is happening? These vacuoles in organisms like amoeba, it is helped in storing the food that whatever is consumed, whereas in some it is also used for expelling excess water and some waste from the cell. Now let's go to the last topic of this chapter, that is the cell division. So new cells are formed in organism in order to grow, to replace old and dead injured cells, and to form gametes required for reproduction. So new cells or cell division is very important. Why? Because some cells are very old and their functioning has probably stopped, so you need to replace them. Some cells may be dead, some may be even injured. So in that case, these new cells are required so that there is a continued production of new cells. So the process by which new cells are made is called as the cell division. Now there are two major types of cell division: mitosis and meiosis. So in mitosis, the process of cell division by which most of the cells divide for growth is called as mitosis. This cell division is mostly related to the growth of the cells. Now in this process, each cell called a mother cell, it divides to form two identical daughter cells. So there is one mother cell which further divides to form the two identical daughter cells. When I say identical, they are going to be same, and therefore they will have the same number of chromosomes as the mother cell. Both of them will have same number of chromosomes as the mother cell. So therefore it helps in growth and in repair of tissues in organism. Now talking about meiosis. So specific cells of reproductive organs or tissues in animals and plants, they divide to form gametes which after fertilization give rise to the offspring. Now they divide by a different process that is known as the meiosis. Now this meiosis, it involves two consecutive divisions. When a cell divides by meiosis, it produces 4 new cells. In mitosis, it was producing 2 new cells, and in meiosis we are getting 4 new cells. The new cells have only half the number of chromosomes than that of the mother cell. So in case of mitosis, the mother cell and the identical daughter cells were having the same number of chromosomes as the mother cell. If the mother cell is having 32, then mitosis both the daughter cells will also have 32. But in this case, what is happening is that each of them is going to have half the number of chromosomes. Understood? So in that case, we can say that if we are talking in division terms, say if the mother cell has 32, then the daughter cell we will have only 16 of it. So this is the difference between mitosis and meiosis. So that was it for today's chapter. Please don't forget to like, share and subscribe to Bodhi Academy.